The cell cycle describes a cell that is programmed to divide. When a cell does not divide, it leaves the cycle. It's called the G0 phase. Cells are not dividing. But when cells are on track to divide, they follow this cycle. What you see in this gray color is most of the cell's life. This is called interphase. Interphase is the cell just growing, doing what it's doing, making proteins, whatever its job is. But then this short little M phase is the mitosis phase, which we'll see on another slide. That's when the cell is actively dividing. Interphase, when the cell is not dividing, it goes through 3 subphases. The initial G1 phase, just think of a cell that divided. The 2 new cells are going to be pretty small. So this first interphase, subphase G1, this small cell that just divided, is going to start growing as it gathers nutrients. Then the S phase is the phase where the DNA replicates, because it's getting ready for mitosis. Remember, in the mitosis, each new cell will need a complete copy of the 46 chromosomes, so the DNA has to be replicated. That happens in interphase, specifically during the S phase of interphase. Then we have another growth phase, G2 of interphase, to prepare for mitosis. Some organelles will divide so that each new cell gets the appropriate amount of organelles. So interphase is divided into 3 subphases, G1, S, and G2. Interphase is when the cell is not dividing but is preparing to divide, where M phase is when the cell is dividing. Some cells that don't divide, that always stay in the G0 phase, that is what this question is asking here, are neurons, which are nerve cells, and muscle fibers, which are skeletal muscle cells. Chromatin, which is the long thread-like version of DNA. You might recall I asked you to think about pulling a thread from a sweater and how long it would be if you kept going until the sweater was gone. But the chromatin, that long thread-like version of the DNA, condenses. It packs together really tightly to form chromosomes during mitosis. So during interphase, G1, S, and G2, DNA is in chromatin form, but during mitosis, chromatin condenses into chromosomes, and that's the form of DNA during cell division. Each chromosome consists of sister chromatids. So I'm going to explain this. Recall during the S phase of interphase, we replicated the DNA, we copied the DNA in preparation for mitosis. So these are 2 copies of the same chromosome. They're referred to as sister chromatids. So when you see this word chromatids, it's when one chromosome is paired with its copy. Where they come together at this region right here, that's called a centromere. Mitosis consists of 4 major phases. Think of the acronym PMAT, P-M-A-T. Don't worry about this phase right here. PMAT stands for prophase, metaphase, anaphase, telophase. So during P, or prophase, the chromosomes condense, just like we saw on the previous slide, so they're visible for the first time in the nucleus. We can't see chromatin. Chromatin's too thin and thread-like. But when the chromosomes condense during prophase, they are visible in the nucleus. But the nucleus starts to disintegrate. Consider this continuation of prophase, and you can see the nuclear membrane starting to break apart. The centrioles These are organelles that were introduced earlier in the chapter. The centrioles begin to produce these spindle fibers, which start to migrate towards the opposite poles of the cell. That's prophase. During metaphase, all the chromosomes with their copies, their sister chromatids, line up in the center of the cell, and the spindle fibers are going through these chromosomes. During anaphase, the copies pull apart. So now we have a set of chromosomes being pulled toward one end of the cell, and their copies being pulled toward the other end of the cell. So anaphase is a quick phase. It's the movement phase. Begins when these chromosomes split apart and ends when they reach the ends of the cell. Telophase can be considered opposite of prophase because the nuclear membrane reappears. Now we have two nuclei. The spindle fibers start to disintegrate, and the chromosomes that are condensed will start to unwind to become chromatin again. So mitosis is the process where the nucleus and the genetic material within it divide. But what happens simultaneously, it's just shown in a different column here, is a process called cytokinesis. Cytokinesis is where the cytoplasm starts to pinch in. This is called a cleavage furrow right here, which divides the cell into two. Cytokinesis actually begins in the late part of anaphase and continues through telophase. Here's a good summary slide. Mitosis is division of the nucleus. Cytokinesis is the division of the cytoplasm. Cytokinesis begins in late anaphase and ends at the end of telophase. Remember that DNA replicates during interphase, not during mitosis. More specifically, DNA replicates during the S phase of interphase. Cytokinesis overlaps mitosis, and I mentioned this a minute ago. It begins during late anaphase and continues through telophase. Sometimes new terms start running together, so this is just a slide to be careful not to confuse some structures with other structures. I'll just run through these. Chromosome, it's a form of DNA during mitosis when it's condensed. Chromatin is the form of DNA during interphase when it's unwound and thread-like, where a chromatid is when chromosomes are paired with their copy during mitosis, and we call each pair of chromosome a sister chromatid. So don't confuse these three, which are very similar with each other. A centriole is that organelle that's needed for cell division, and they come in pairs. A pair of centrioles is called a centrosome. Now that word sounds similar to this one, a centromere. A centromere is an area where two sister chromatins are bound. And if you go back to the same point of this video where I look at the centromere, or where I describe the centromere, you'll see a kinetochore is labeled there. A kinetochore is where the spindle fiber, sometimes called the mitotic spindle, attaches to the chromatid.